talk. So our next presenter is Associate Professor Andrew Zalesky, and his talk is titled Circuit-Based Personalisation of TMS with Neuroimaging. So Andrew is an Associate Professor in Engineering and Medicine at the University of Melbourne, and he is an internationally recognised leader in the field of brain connectomics. He's ranked in the top 1% most cited researchers worldwide in the area, and is also the developer of the network-based statistic tool for testing hypotheses about the human connectome, which I'm sure some people here have probably used in their own research. So I'll pass on to Andrew to present.
Thanks, Aaron. It's wonderful to be able to present today and to share some of our group's work here in the area of TMS and neuroimaging. I'd first like to thank the executive committee for inviting me to present today, and as well as Aaron for coordinating the meeting. It's wonderful to see that such an important area now has representation and advocacy in Australia. So really congrats to the President, Professor Kate Hoy, and the executive committee for setting up the society. And I really hope to be able to contribute and participate in the society going forward in coming years. So my background's in electrical engineering and neuroimaging, and what I hope to present here is approaches that we've used to understand the impact of TMS on brain networks using advanced brain imaging techniques. And in the second part of the presentation, I will turn more to clinical applications and using brain imaging to personalize and optimize TMS therapies, particularly in depression, but also some recent work that we've been doing in OCD. So to begin, I just can't move on to my next slide. There we go. I won't spend too much on giving you a rundown of TMS. I'm sure everyone is familiar with the technique. I will say, however, that the methods that we present here are fairly general and can be applied to other brain stimulation modalities. I won't talk about that today, but these principles are general, and we feel that these can be applied to other techniques. So I'm going to focus on two particular kinds of brain connectivity. And for those that are not familiar with connectomics, I'll present a quick overview of those two connectivity measures. One is functional connectivity, as I've shown here on the left. And so in several of my slides that you will see a functional connectivity map as I've shown here. And this is always relative to a seed region. So in this case, the seed region is the PCC. And what we do, we can measure with fMRI the neural activity at that region. And for each other voxel or pixel in the cortex, the color of that pixel represents the extent of synchrony in the activity between the PCC and that region. So for example, we can look at the medial prefrontal cortex, and that's colored with warm colors, red colors. And so that means that there's positive synchrony in those two areas. When the PCC goes up, so does the MPF. When the PCC goes down, so does the MPF. Conversely, the cool colors, the blue colors here, such as the IPS, they're anti-correlated with the PCC. So in this case, when the PCC goes up, the IPS goes down, and so forth. The other kind of connectivity that we can measure using neuroimaging techniques is structural connectivity. This represents the physical, the axonal fiber bundles that interconnect brain regions. And so these can be measured using a modality referred to as diffusion MRI and an algorithmic process called tractography. I won't spend a lot of time on structural connectivity in this presentation. I wanted to begin with emphasizing the importance of characterizing the network-wide effects of focal stimulation, such as TMS. A lot of work is focused on understanding the local effects in the vicinity of stimulation. This is a study, though, this is one of the first studies actually by LDF that characterized network-wide effects. So this is a TMS, a 20 Hertz TMS study where individuals were placed in the scanner immediately before the stimulation. And I'm showing here a functional connectivity map from the stimulation site, which was the inferior parietal lobule. After they came out of the scanner, they were stimulated for 22 minutes. at 20 hertz, and then they were put back into the scanner, and a second functional connectivity map from the stimulation site was constructed, as I've shown here. And so we have pre and post, and we can then subtract pre from post to come up with a difference map. And crucially, we see differences in connectivity that are distant from the focal stimulation site. Moreover, that some of these regions are not structurally connected to the focal stimulation site. In some more recent work with Luca Cocchi, we've looked at this with theta burst TMS. In this case, we stimulated two regions. We stimulated the visual cortex, as I've shown there on the left, and we also stimulated a hub region, in this case, the frontal eye field. So the hub is a region that's widely connected with other regions. And our hypothesis was that the hub region, stimulating the hub region, would result in more extensive, more widespread effects that were spread across the cortex. But that's not what we saw. In fact, we saw that stimulating the visual cortex resulted in increases in connectivity, as depicted here with the warm colors. In contrast, stimulating this hub region, the FEF, resulted in decreases in connectivity. And we weren't quite sure how to interpret this. What was the underlying mechanism that was resulting in either a decrease in connectivity for the FEF and an increase for the visual cortex? So to address that question, we turned to biophysical modeling. And the basic premise here is that the underlying structural connections that comprise the human connectome allow the effects of stimulation to propagate, to transmit to downstream regions. And these are the effects that we can detect using functional MRI. Now, we can map those structural connections, those axons, using diffusion MRI, as I've already mentioned. And we can do that at an individual level. We can take a person's diffusion MRI scan, map their connectome. And once we've done that, we can then develop, or then use a biophysical model, as I've shown here on the right, to simulate in silico the effect of TMS across the whole brain. So in this model, each region is characterized by a population of excitatory and inhibitory neurons. That's the red and the blue circles here. This is a neural mass, a so-called neural mass. And each of these regions, each of these populations of exhibitory and inhibitory neurons are interconnected based on an individual's structural connectome, so the data that we get from the diffusion MRI. Now, the advantage of this approach is that once we have the model set up, we can systematically evaluate the effects of stimulating different regions using different pulse designs, different protocols, and so on and so forth. So it's a very rapid and inexpensive way to comprehensively evaluate the impact of stimulation when we're changing the stimulation target or the protocol. So in this particular model, we have the ability to change the ratio between the excitatory and the inhibitory population of neurons at the particular region that we're stimulating. So in this image, it's a frontal region. But of course, this can be changed rapidly just by changing a parameter in our computer code and rerun the simulation and understand what's happening at a subsequent region. So you might ask, well, okay, you have this model, but what does it actually tell us? What have we actually found from this? And so this is some work from Leonardo Golla, who's now at Monash. It was a couple of years ago where Leo systematically evaluated 12 candidate sites and then mapped the effect of stimulating these sites or the changes in connectivity across the cortex. I won't go into the details of this, but one of the key insights that came out of this study and some of the other studies is that we see that there is an intrinsic frequency associated with each brain region. And so the sensory regions, those that are low in the hierarchy, generally oscillate very fast. They have a fast intrinsic frequency. As we move up the cortical hierarchy to frontal regions, such as OFC, PFC, and so on, the intrinsic frequency slows down. And so this enabled us to explain that initial finding where we were stimulating the hub region, the FEF, and the sensory region. So what happens if we use inhibitory type stimulation and we stimulate one of these fast regions, such as the sensory region, that's going to slow down the intrinsic frequency and therefore bring it closer to the other regions, particularly the higher order transmodal regions. And that's why we see an increase, or that's why we think we see an increase in functional connectivity when we stimulated the sensory cortex, the visual cortex. And conversely, when we stimulated the FEF has a slow intrinsic frequency. We're going to slow that down even further and therefore take it further away from the intrinsic frequencies of other brain regions. And that's why we think we see the decrease in connectivity. You might be asking, why is it important to understand the network-wide effects of brain stimulation, such as TMS? And this is a really nice study from Michael Fox. It came out 5 or 6 years ago. What Fox did here, he considered a range of disorders which show efficacy to treatment with both DBS and TMS. So if you focus here on the case of anorexia, which is on the top right, what Fox did is that he considered the DBS site. He mapped a functional connectivity map from the DBS site, and that's shown on the right here. So once again, the warm colors represent regions that are in sync with the DBS site, and the cool colors represent regions that are out of sync, that are anti-correlated. And crucially, what Fox then did, he overlaid established TMS sites for each disorder. And remarkably, what we see is that the TMS sites overlap very well with hotspots, or with regions that are either colored, that are either high in sync or that are out of sync with the DBS site. So the crucial conclusion is that when the therapeutic efficacy of brain stimulation is not relative to a single focal target, we're rather modulating a network of regions. And so it's important to consider the effect within this broad network and to consider therapeutic efficacy relative to the broad network, as opposed to considering the specific region. I'm now going to change pace and move to some of the work that we've done in using neuroimaging techniques to personalize and optimize TMS therapy for depression. This is work that has been led by Robin Cash. Robin Cash is a DECRA fellow in my group at the University of Melbourne, has really done some wonderful work in personalizing TMS for DLPFC and using advanced functional MRI techniques to do so. What I've shown here is a timeline from one of Robin's recent reviews in this area. Check out the review. It's an informative review in the area of in TMS and personalization. And it's important to note that personalization isn't a new area in TMS. In fact, for those that are working with DLPFC, TMS, the so-called 5 centimeter rule, in some sense is a crude form of personalization. As time has progressed, we've now moved to a stage, as I've shown here, or as Robin's shown here in panel F, where we can now use functional MRI and functional connectivity to guide personalization. To clarify what we mean by personalization, personalization here is really where stimulation is optimized to suit an individual's brain structure, their function, connectivity, and or possibly their clinical symptoms. And this is where we can potentially use approaches such as biotyping that Colleen touched on in her presentation. Here, we've only focused on personalizing the stimulation target site, but it's also important to note that personalization can be performed in terms of optimizing the stimulation frequency, the protocol, and so on, as well as patient selection. But this work will only focus on the target site. So moving now specifically to depression and DLP and the dorsolateral prefrontal cortex, which is an established TMS target for depression. We know that we should be, the spatial extent of the DLPFC is large. And so the question is, what particular area or region of the DLP should we target? And there's clinical evidence, as I'll present in a moment, as well as some theoretical arguments for targeting a region within the DLPFC that is anti-correlated with the subgenual cingulate cortex, the SGC. So what I've shown here is a functional connectivity map with respect to the SGC. This was computed in 1,000 healthy individuals. These were taken from the Human Connectome project. And as I've mentioned before, the red areas in this map are regions that synchronize with the SGC. So when activity in the SGC goes up, so does the red regions. In contrast, the blue regions are regions that are out of sync with the SGC. So once we had this map, the idea is that we should target the optimal target coordinate is somewhere within this blue region, the region that's anti-correlated with the SGC. And what Robin did, this is work with Professor Paul Fitzgerald. This was in a retrospective study where we looked at 24 individuals with treatment-resistant depression. They received daily 10 Hertz RTMS to the left DLPFC using the F3 beam approach. Treatment was for about 3 weeks. And what we were then able to do, the F3 beam approach, the actual coordinates do show inter-individual differences. And so the grey dots in this image show the specific location of stimulation for individual patients. I'm not sure how clear those grey dots are, but you can see these dots over the DLPFC for the FC map shown on the right-hand side of the slide. And what Robin was then interested in saying is, well, in testing was whether if serendipitously the stimulation target was located at one of these areas of the DLPFC that was anti-correlated with the SGC, would treatment response, as measured with percent improvement in MADRS, would treatment response be improved? And so as we see with the correlation on the bottom, that was indeed the case. So the x-axis here is the functional connect. So each dot here represents a patient. The x-axis is the functional connectivity for that patient from their actual stimulation site to the SGC. And the y-axis here is the percent improvement in MADRS symptoms. So the more anti-correlated, the more negatively correlated the actual stimulation site was to the SGC, the better the treatment response. Crucially, it's important to remember here that the FC map that we're using is a group average from healthy controls. It wasn't computed from the individual patients. And the problem with that, as I'll show now, is that we see that there is marked variation in the functional connectivity architecture of the DLPFC across individuals. So here I've shown 4 particular, if you look at the top row, this is 4 particular individuals. And we can see that the candidate target or the desired target, the blue region, varies markedly across the spatial extent of the DLPFC. So for this individual, it's more lateral. For this one, it's more posterior, and so on and so forth. You might say, oh, well, this is just because there's noise in your fMRI scan. So this variation that you're seeing across individuals 1 to 4 is due to noise in the scanner. It's not real variation. That's not the case. We actually took a second scan in these individuals approximately a week later, and we can see that there is correspondence between scan 1 and scan 2 in each individual. So in particular, the greatest, the location of negative correlation shows spatial correspondence, and that inter-individual variation still exists. So this points to the importance of personalizing the stimulation site to suit an individual's DLPFC functional architecture. So what we're proposing here, this is a personalization approach where before treatment, we have a 20-minute functional, at least a 20-minute functional MRI scan. We've then developed a software tool that we call OptiStim. This is work that Robin Cash has led. And the functional MRI scan is fed in to the OptiStim software. And the OptiStim software will then output a coordinate, a stimulation coordinate that is optimized to the functional connectivity map of the individual patient. So we then went on and tested, well, perhaps I first say a few words. I mean, I don't have so much time about the software tool, about OptiStim. There is some challenges in mapping functional connectivity maps at the individual level, at the individual patient level. And so we've implemented some particular strategies, methodological strategies to ensure that we can reliably and accurately compute the optimal stimulation coordinate. I won't go into the details here, but we can, what I will say is that we can, across different sessions, we can locate the optimal coordinate with a precision of around about ±3 millimeters, or in some cases better. That's the average performance across a group of healthy subjects. One of the crucial considerations is the length of the fMRI scan. We're advocating for at least 20 minutes. For shorter scans, computing a reliable target becomes more challenging. And so this is work that's currently in revision at Human Brain Mapping that Robin has led. Once we had OptiStim developed, we wanted to go on and test in a clinical setting whether personalisation offered any advantage. And we've done this in a retrospective study. This is once again work with Professor Paul Fitzgerald. This is the cohort of 26 individuals with treatment-resistant depression. It's the same cohort that I described earlier. They received 10 hertz of RTMS, the left LPFC. It was over a 3-week period. In each week, they attended the clinic 5 days a week. On the right there, we see clinical response. So we see that there is the distribution of response is almost bimodal. So there are some individuals that responded quite well, others didn't show a response. And so those that didn't respond, perhaps they stimulation wasn't targeted to an optimized region. So here we used, Paul used the F3 beam approach for stimulation site selection. Once again, in this study, there was a pre-TMS as well as a post-TMS fMRI scan that sat in at either ends of the stimulation treatment. So what we tried to do is we tried to apply our OptiStim software retrospectively. And so I've given this diagram to give an understanding of what we've done here, is that we've taken the pre-TMS scan. In fact, what we did, we combined the pre-TMS and the post-TMS together. But for the sake of presenting this, let's just think that we take the individual's pre-TMS scan, we input that to our OptiStim software, and that retrospectively computes the optimal stimulation site for that individual, as I've shown here on the map, the functional connectivity map on the bottom. We then, during the actual TMS session, the precise stimulation, the actual stimulation coordinate has been recorded, and we reconstructed that and projected that onto the individual's brain as well. So we have both the optimal coordinate and the actual stimulation coordinate that was derived from the F3 beam approach. What we've then done is measured the distance between those two coordinates, the optimal coordinate that we've computed retrospectively and the actual stimulation coordinate. And we did that for each of the 26 individuals. And so for some individuals, that distance is quite large. For other individuals, serendipitously, the actual coordinate has actually fallen right on the optimal coordinate, and therefore the distance is short. What we hypothesized here is in cases where the distance was short, the clinical improvement would be superior relative to patients where that distance between optimal and actual was large. You might argue, or a skeptic might say, well, how do you know you're not just that your OptiStim software is not just predicting the same coordinate for each individual? So you're not just predicting a group coordinate and there's no individual variation. We do, however, see, to respond to that criticism, we see quite substantial variation in the optimal coordinate across individuals. So here's the results of that retrospective evaluation. We did indeed find, this is work that was just published a couple of weeks ago in JAMA Psychiatry, we did find a strong association, a strong and significant association between clinical improvement in MADRS, percent improvement in MADRS, and the distance between the retrospectively computed optimal site and the actual site. So each circle here represents a particular patient. The x-axis is the distance that I just described, and the y-axis is the percent improvement. So this provides retrospective evidence of the potential effectiveness or the potential improvement that can be achieved by personalizing the stimulation target based on functional MRI connectivity. Crucially, we also ran a partial correlation analysis where we controlled for the distance to the group average DLPFC target. So the group average was computed based on a normative functional connectivity map. And we still saw a significant correlation after controlling for this distance. So this accounts for the argument or for the possibility that OptiStim, our software, is not just computing the same target, the same group consensus target for each individual. Robin also considered a range of established TMS targets for the DLPFC, and we did not find any association between treatment response and the distance to those so-called the non-personalized or the consensus targets. To wrap up the talk, I'd just like to briefly focus on some of the work that we've done more broadly in the area of personalization. This is not specifically in the area of TMS. This is some work that we've done to parcellate the subcortex, and so this is crucial. Having an atlas of parcellation for the subcortex is crucial in terms of identifying targets. And so this is work that was done by Yet Tian, a PhD student in my group. It was published in Nature Neuroscience a couple of weeks ago, or about a month ago now. It's a multi-scale atlas. It's a hierarchical atlas where we have a subcortical nuclei at the core, and then we gradually get finer and finer parcellations. We see that the boundaries between regions change according to the particular context or the task that a person is engaging in. And the main reason that I wanted to show this work or present this work on the subcortex is that the architecture, the regions of the subcortex vary substantially across individuals. So at the top here, I've shown the example of the striatum. I've shown the group consensus parcellation of the striatum, and then I've shown two subjects. One that agrees quite well with the group consensus, that's the middle row here, as well as a subject that shows poor consensus or deviates substantially from the group consensus atlas. Apologies. So this is another representation of that for the hippocampus, where we've got a particular subregion of the hippocampus, and I've shown the distribution of individuals, how well they overlap with that group consensus. So we see that the majority of individuals show 80% overlap with that consensus parcellation. But if we move to these particular individuals that are out in the tail, the overlap is as low as 20 or sometimes a little high, up to 40 or 60%. So this points to the importance of personalizing. It gets back to the idea of personalizing site. There are many individuals that will conform to the group average, but those individuals that don't respond to therapy, that might be because their functional architecture, their brain anatomy deviates substantial from the consensus, and therefore they may benefit from personalizing the stimulation target or other aspects of the stimulation protocol. In the interest of time, I'll skip over that and move to my conclusions and a summary. So in summary, I hope I've convinced you of the importance of characterizing the brain-wide effects of stimulation. I think it's important to characterize therapeutic benefit, not in terms of a focal region, but a network of regions and modulating that network of regions. I think that computational modeling is going to play a more important role in evaluating and prototyping stimulation protocols. It's a very efficient, rapid, and high throughput approach for assessing the effect of changing the stimulation site and aspects of the stimulation protocol. Of course, there are disadvantages to computational modeling, and there's always a need to evaluate those results and validate those results using in vivo techniques, as I'm sure Bedita, our next speaker, will discuss. I presented our OptiStim software. So this is work that was, this is software that was developed by Rob and Cash. So this allows us to take an fMRI scan and compute the optimal DLPFC coordinate at an individual level. And I presented recent retrospective evidence pointing to the clinical efficacy of target personalisation. It's important to emphasise that this is retrospective. We still need to run or perform a clinical trial on personalisation and to evaluate OptiStim. I would also like to mention that we do have some clinical trials that are running on TMS in OCD. This is work with Paul Fitzgerald as well as Luca Cocchi, and hopefully we'll have some results in this area in the coming years. And one thing that I haven't mentioned here is structural connectivity. I also think that's an important way forward in terms of not only looking at optimization in terms of functional connectivity, but also integrating structural connectivity. I think that plays an important role. Acknowledgements. The work here on personalization has been driven by Rob and Cash. He's really been instrumental to the work that I've shown here in personalization of the DLPFC and development of OptiStim. Luca Cocchi, who's also been has contributed to both the modeling work as well as the personalization work, and he's currently running a trial of this in OCD. Leo Gollo, who's at Monash University, he's involved in the computational work, and of course, Professor Paul Fitzgerald, who's, as I'm sure you all know, is an internationally renowned expert in the field of brain stimulation, who's been involved in all of these studies as well. Finally, I'd just like to, a bit of an advertisement is that we're running the Australian School of Connectomics towards the end of next year. If you're interested in learning about brain networks and approaches for studying brain networks, consider signing up. We'll probably make an announcement either early or mid next year about the school, and it will probably run for 3 days. So that's all from me. Once again, I'd like to thank the Executive Committee, Kate Hoy and colleagues, for inviting me to present. And I'd be very happy to take any questions.
Thanks so much, Andrew. Really great talk, really interesting stuff. It seems like these personalised approaches are definitely the way forward with a lot of this work. We'll just see if we've got any questions. If people want to ask, we should have time. We've got one here in the chat from, oh, sorry, did someone have a question?
I was just saying, yeah, I've got a question.
Yep.
Thanks, yeah. Oh, and thanks everyone. This is like one of the most fascinating Zooms I've been at in this entire unusual year. I really appreciate it. Andrew, in that retrospective look at the distance in centimeters, millimeters versus treatment response that happened, and I realize you might, this is a bit specific, you might not.
I can go back to the slide here, perhaps, if that would help.
Thank you. Yeah, that'd be good. Oh,
sorry. I think I've. Aaron, can I share the slide again?
Yep, you should be able to. Yep.
Let's see if I can get back to the. Is that the one that you were referring to, Andrews?
Yeah, keep going back. One more. Back one more.
That one?
There, thank you. I was wondering why it was the metric chosen was physical distance, and why, or was it run also with strength of correlation or anti-correlation at that point?
Yes,
because you can go 1 centimeter and you might be in a red zone in one direction, but you might still be in blue in a different direction at a further distance, for example.
Yeah, that's an excellent point, and it's one of the things that we discussed, whether the metric of most interest here is distance or connectivity, functional connectivity itself. And we actually replicated this result with functional connectivity. I haven't shown that here, but we can get to the same result by using, rather than using the distance itself, using the actual functional connectivity between at the optimal and the actual site. I think the correlation is more or less the same. If you're interested, there is the we report that in the paper. But it's an excellent point, and I think there's the question there also of whether you make a good point, is if we move a certain distance, that we could go from a red to a rapidly transition from a red to a blue region, and that might not be a distance might not be the best metric to capture this. And so when we first did these results, when Robert first looked at this, we did look at distance, and this came up, and we replicated this using the actual functional connectivity value.
Right, thanks. Yeah, I'll take a look. So yeah, it didn't actually, but it didn't actually change the strength of the relationship that much. You said it was similar.
I don't have the correlation I mean, if Robin's here, he might be able to chime in with the exact correlation coefficient. I don't know it offhand. It is reported in the paper.
I'll go take a look. Thank you. Thanks for answering that.
Anyone else?
Hi, Andrew. It's Manrina Kaur here. I did put my question in the chat, but I can just read it out to be more interactive. So thank you for a great talk. It's really great to hear about the developments in individualized functional connectivity maps for this purpose. Obviously, this has been quite a big hurdle until this point. So I'm really keen to hear a little bit more on how you've overcome some of the difficulties in generating reliable functional connectivity maps for each individual. So one of the issues I've known about is the signal to noise ratio of the subgenual ACC. That's just one thing, but yeah, just really interested to hear on what strategies you've used.
Yeah, that's an excellent point, and I think one of the greatest challenges here is in measuring functional connectivity at the individual level, particularly for subcortical regions and regions such as the SGC. They're small, and as you've mentioned, the signal to noise ratio is low. I would first say that one of the most important things is the length of the acquisition. Having a long acquisition is crucial. In fact, in this study that the retrospective, we only acquired 7 minutes. And so what we did, we combined the pre and the post fMRI to increase the length of the acquisition. One of the risks, of course, is that the actual TMS could have changed activity in the post, and we tested for that. We didn't see any changes, so that gave us confidence to merge the two, to concatenate the two scans. But even 14 minutes is relatively small. So we have got, if you check out that HBM paper, we've actually measured the reliability as a function of time, and so that's a crucial factor. The other factors are, there are approaches such as what's referred to as the seed map approach. I don't know if it's necessary to go into the details here, but it's a way to boost the SNR of small regions rather than actually using the activity within the region itself, such as the subgenual cingulate. We can compute a network from the SGC to the rest of the cortex and, well, essentially use the activity from the rest of the cortex and thereby boost the SNR rather than just using the SNR within that small region. There's also acquisition. There are ways that we can improve acquisition. So if we're measuring subcortical regions, there are tweaks to the actual acquisition protocol that can be done to improve signal to noise ratio for these subcortical regions.
Yeah, great, thank you. I'm looking forward to reading that paper. I know one of the strategies is to use a seed in the limbic region, which is maybe what you were referring to when you were talking about the network.
Yes.
Yeah. Okay, thanks.
Great. I think in the interest of time, we might push on. I can see Robin Cash has also put some information in the chat as well regarding that previous question too. So if anyone's interested. All right. So I think we'll go on to our final presenters. Thank you again, Andrew, for that really interesting talk. It was great to hear.